it took that long to get FDA approval for a treatment that we knew worked. So this kind of illustrates the challenges in developing new treatments. And then looking way back where we started, it's always good to cite Sir Archibald Garrod, who had described alkaptonuria, albinism. Other Wikipedia misspells albinism, but still generally the information's good. And that expanded to over 5,000 disorders that could fit this description, this category, in the online Metabolic and Molecular Bases of Inherited Disease, for those who remember that textbook. And importantly, each is linked to an enzyme or protein deficiency. So we have this kind of paradigm with the pathway, missing enzymes centrally, and then accumulations of substrate for that enzyme, and deficiency of products. But the key thing in this context, I believe that the most effective treatment for these disorders is to replace what's missing. And that's the goal for current gene therapy. Shifting gears then into a little background on gene therapy. Typically, but not always, I'll give examples of other approaches, but typically it's a modified virus. And this removes viral genes. It just keeps essential components needed to deliver a therapeutic gene termed a transgene. And then it's important to think about how there could be a response to that viral vector. And this response can either be to the vector part, the viral part, like an AAV vector particle shown on the left, or to actually what it's delivering, the transgene. And furthermore, it could be potentially related to the parts of the vector genome that are from the virus, or the transgene within the vector genome, which is the therapeutic payload. So these are some of the basic components for a viral vector used in gene therapy. And very broadly, we have two approaches. One is a direct infusion of the vector intravenously, or more targeted to a particular tissue, but an injection, like a typical intravenous medication. Or two is to inject a treated cell, like a stem cell, that would contain the transgene, and then those cells would, go into the body and graft, and those would be the source of the therapeutic effect. For the infusion approach, injecting the gene therapy, typically adeno-associated virus, or AAV, is used. It's a non-pathogenic parvovirus. Importantly, it's not adenovirus, but a parvovirus. It transduces dividing, non-dividing cells. Typically, it doesn't integrate. It sits in the host cells in the nucleus without integrating the chromosomes. That's important. But the transgene can be sustained. There can be long-term benefits from these episomal AAV vector genomes. For the stem cell approach, often a lentivirus is used to integrate in the stem cells, and then the cells are delivered. These are often derived from HIV, but the HIV is completely inactivated, so it can't cause disease. This too will transduce dividing and non-dividing cells, but it's also not efficient at transducing cells following just a simple injection in the body. That's why it's used to treat cells outside the body and then deliver the cells. It is integrating, so it's stable within those delivered cells, and transgene expression is typically sustained. So any benefit should be stable. With regard to AAV, again, our favored viral vector. It's very small. It works like a nanoparticle, 20 to 25 nanometers diameter, so it can escape through endothelial cells from the bloodstream into tissues. It also has a fairly small genome. It just holds 4.7 kilobases approximately, including the AAV parts needed for packaging as well as the transgene. So the limit of the size of the transgene to be delivered is about 4 kb. And that's an obstacle for some diseases, not the ones I'll talk about today. AAV is dependent on helper virus. That's where adenovirus comes in, because adenovirus provides helper functions to be able to package these AAV particles. But by itself, AAV is completely replication deficient. It isn't copied. It isn't a productive infection. And also, there are many different types of AAV that differ in the AAV capsids, those proteins that package the transgene, the vector genome. And then if there happen to be antibodies against one serotype of AAV, that won't work in that, that host who's essentially been immunized. But potentially one could use a different AAV in that same patient, so long as they didn't have antibodies against that other serotype of AAV. You could see also in the table in the lower right that having these different serotypes gives us more options in terms of directing the transgene to different tissues. So for instance, AAV8, AAV9 are good in liver, and for instance, AAV1 is better in skeletal muscle. So some have termed AAV vectors as kind of a toolkit that can be used for different kinds of projects. Now, talking about transduction. So transduction is actually the term for this AAV vector to deliver the transgene to a cell. And there are many steps. It's incompletely characterized, but we know that it involves binding in the cell membrane, uptake through endocytosis, trafficking to the nucleus, and then uncoding, removing those capsid proteins from the DNA. And that, that DNA is a vector genome that needs to go from single-stranded DNA to double-stranded DNA because AAV starts as a single-stranded DNA, and you need double-stranded DNA for gene expression. And then the AAV will concatamerize. It'll form end-to-end copies in an episomal form, and it'll sit in these cells and express the transgene. Only a very small fraction will integrate into any chromosome site, and there's no preference where it integrates. It could be completely random throughout the genome when it does integrate, and that frequency is estimated at less than 1%. So if we shift gears then to some examples of gene therapy, and here's an experiment that my lab did a number of years ago in collaboration with Carrie Harding. Actually, we made the vector and Carrie did the experiment. It was for treating PKU in mice, and the deficient enzyme is phenylalanine hydroxylase, and we replaced that with an AAV8 vector. And doing that, we intended to lower the phenylalanine and toxic metabolites that accumulate in PKU and then produce tyrosine, which is an essential amino acid in PKU in absence of PAH expression. So here are the mice, and like untreated PKU patients, these mice have an abnormal pigment of their fur. They don't have full pigmentation. This is related to the tyrosine deficiency. And once treated, then the mice developed a darker, more normal coat color. You can kind of see indicated by the arrow here in the picture. Down in the graph, we have a very high phenylalanine concentration, over 2,000 starting out. And so these mice have high Phe, just like patients. And that came down very nicely after delivery of this AAV8 vector to make PAH. And then gradually over the following weeks to months, the Phe level crept up again. And this shows how, for this episomal vector, at least in some experiments, it will be lost. This has to do with cell division in the liver, treating a very young animal. And then as the liver grows, the mouse grows, and the cells divide, those episomal vector genomes are lost. So at the end of the experiment, after months, the phenylalanine was higher and the PAH was low in liver. And it wasn't related, at least it wasn't detected that there was any immune response against the PAH that we introduced. But it was a good initial concept for being able to treat a disease like PKU. And this was entirely liver-based. Now moving on to hemophilia as an example, and this is much more developed. Hemophilia, of course, is a fairly common congenital bleeding disorders among the inherited coagulopathies. And it's characterized by frequent bleeds, especially in the joints, can cause very severe complications, especially when bleeding is in a closed space, like in the joints or like in the brain. It currently is treated by periodic injections of the coagulation factor that's missing, factor 8 or factor 9. Here is the coagulation cascade, the intrinsic pathway, extrinsic pathway. You can see how factor 8, factor 9 are central here. Factor 8 is deficient in hemophilia A, factor 9 in hemophilia B. Both are X-linked recessive, and so about 1 in 5,000 males have hemophilia at birth. And then importantly, there's a range of severity, and less than 1% of normal coagulation factor will cause a severe disease. And then as little as 1 to 5% factor will lead to moderate or more mild symptoms. So with gene therapy, not very much factor needs to be produced to improve the symptoms. So this is a good example and a good target for gene therapy. Here's a clinical trial led by the company Spark a few years back in hemophilia B, and they used actually a factor 9 variant known as factor 9 PADWA. It's naturally occurring. Patients who carry it have increased clotting, so it's a super active factor 9. There was an advantage to use this for gene therapy because less of the factor 9 protein would increase clotting or treat the symptoms more easily. The dose was low here, so this terminology is 5 times 10 to the 11th vector genomes or transgenes per kilogram body weight. And I'll tell you, this is a low dose and it's unlikely to cause complications. They chose patients with a low factor 9 level, so they had severe hemophilia. They also were screened for not having anti-AAV neutralizing antibodies. The idea is a patient who had been exposed to AAV, had neutralizing antibodies, wouldn't benefit from this gene therapy, so screening for that. And here are the data. At the end, they had up to 30% normal factor 9. You can see in this graph how the individual lines over the weeks following the vector administration show increased in factor 9 for everyone shown here. All but 2 are shown. A couple of them had reduced factor 9. This was related to immune responses. There's a very well-characterized T-cell response against the AAV capsid protein that some patients produce. This is flagged by an elevated liver transaminase, ALT. So when the T-cells are activated, the patient's liver cells containing the vector start to be killed and the ALT goes up. Now there's a treatment for this, and it was developed before this study in particular. The idea is to monitor for elevated ALT, then give prednisone to suppress the T-cell response, and mostly that was effective. Other parts of this that were key were this AAV8 vector going to the liver to produce the factor 9 just there, and that's possible because it's a secreted protein that would then go into the blood and prevent bleeding. This cartoon down here shows actually the design for the vector genome where there's a liver-specific promoter, ApoE hat that is driving the transgene expression. So factor IX only mated liver, it's known that producing the protein only in liver tends to reduce immune responses or antibody responses against that protein. So they had that advantage. The immune response was only against the AAV capsid proteins, not against the factor IX. If we look at then, let's get onto the screen here. A patient who did have the elevated ALT, the T cell response, you can see in the black boxes here, after about 50 days, the ALT goes up. They were monitoring closely twice a week. And at the time the ALT went up, then this participant was dosed with prednisone, starting at 1 milligram per kilogram per day and then tapering fairly rapidly. ALT goes down nicely. And although the factor IX dropped, and this was probably, at least in part, due to loss of some of those liver cells making factor IX, factor IX remained elevated above baseline over the course of this 1-year study, where the patient started less than 2% and ended up close to 20% of normal. Down at the bottom, we can see measuring the T cell responses, and this is an ELISpot assay. It's detecting the cytokine gamma interferon that's released by T cells, actually activated in response to the AAV8 capsid proteins. And it was only present here in the early parts. And then once the prednisone was given and was effective, the ELISpot dropped, showing that the T cells were suppressed. And we can think just a little bit more about immune responses to AAV, because this is an important topic. First off, we've been talking about here in the upper right, so-called cytotoxic T cells that attack the liver cells containing the AAV8 capsid proteins. And we know at the same time that there are antibody responses shown by the symbol 8 here. Antibodies will be made against the AAV once it's been administered in gene therapy. Now, before the study even starts, screening for pre-existing antibodies against the AAV proteins, this anti-AAV8 was done to avoid that immune effect. There could have been transgene immune responses. These would have been against factor IX. Again, the study was designed to avoid that, and apparently they were effective at doing so. And then once the gene therapy is done, we have to realize that the same vector can't be given to the patient again with any benefit, because the antibodies are present now, and they'll neutralize the AAV8 even if it's given again. That's the current situation. There are efforts underway experimentally to be able to eliminate these anti-AAV antibodies, but that's not available clinically yet for clinical trials. Okay, moving ahead then, a well-known study in spinal muscular atrophy. And so, we're aware of type 1 SMA being a very severe and lethal condition if it's not treated. This study out of Nationwide Children's used very high doses of an AAV9. And at those high doses, it's injected intravenously and it actually gets into the nervous system. One immediate observation was the first patient at this high dose had an immediate, within, I think 3 days, elevation of ALT to quite high levels. This was unexpected. It was treated with immune suppression effectively. And then following that, all the subsequent subjects enrolled were given prednisone at the start, a prophylaxis to prevent this immune response. This is then a consideration for the SMA study. So there was an available specific therapy, this ASO, Spinraza, that led to modified splicing, of the SMN1 gene, inclusion of exon 7 that's missing in SMA. And this is this small DNA given intrathecally repeatedly, but it was known to have benefits and already FDA approved. So at the time the clinical trial was done, it was done with sort of unknown potential benefit or toxicities in this phase 1, but it was deemed safe and potentially beneficial enough to go ahead with a study. And the gene therapy works by replacing or introducing a full-length SMN cDNA to make a functional SMN. So the study went ahead, and as we know, there were good benefits. This shows in all but one participant increasing CHOP and 10 scores, and the line at 40 shows where most or any patient with SMA does not surpass that developmental threshold. And again, all but one subject had a significant improvement not expected for SMA. This participant down here, let's see, I think it's patient 8, was treated somewhat later and didn't respond. But so this increase in the intent score led to FDA approval for Zolgensma. Now, given that, there's still unanswered questions. And about the same time, some new experimental data came out showing that with a very similar vector, not Zolgensma, experiments in non-human primates and in piglets actually reviewed some neurotoxicity and some severe liver toxicity. So actual liver failure that hadn't been observed in the clinical trial in the non-human primate model. And then in the piglet model, what are termed proprioceptive deficits and ataxia, loss of balance. And this was related to actually nervous system toxicity. And it wasn't AAV9, but it wasn't Zolgensma. So essentially a red flag and lots of concern. And subsequently to FDA approval, this report from Journal Pediatrics and liver failure out weeks from the actual vector administration, treated successfully with high-dose steroids. But this kind of thing, while it was on the label for the drug, it really illustrated the clinical experience, how some patients were having kind of a severe liver toxicity related to the high-dose AAV9 to treat their SMA. And then finally, preclinically and entirely in mice, concerns that too much of the SMN protein could potentially be toxic. And this was related to SMN overexpression actually in neurons. Again, a mouse model, but both a normal mouse and a mouse with SMA. And this is sort of learning from a clinical trial. It happens often, and it may lead to a second-generation therapy in terms of gene therapy for SMA. I might expect that. All right. So that was the more general part, including clinical trials. And here I'm going to switch gears slightly and talk about work ongoing in the glycogen storage diseases, where my lab has contributed, as well as others. For GSD1A, we know as von Gierke disease, caused by glucose 6-phosphatase alpha deficiency. It causes severe hypoglycemia, initially growth failure. There can be seizures related to hypoglycemia. There are multiple biochemical abnormalities, most of which can be treated and reversed with the dietary therapy, but the dietary therapy consisting of uncooked cornstarch around the clock is not completely effective. And if it's interrupted, of course, the patient has severe hypoglycemia again. And here's the pathway. And replacing the glucose 6-phosphatase enzyme, which is an intramembranous enzyme in the ER, is our gene replacement therapy. Here's the pathway again, showing the biochemical abnormalities related to G6Pase deficiency. As mentioned, there's severe hypoglycemia during fasting. There's hyperlipidemia with extremely high triglycerides, can even lead to pancreatitis, high lactic acid related to accumulating lactate, high alanine, and then hyperuricemia. Later in life, patients can develop gout. So lots of biochemical abnormalities, and you can think of it as having lots of potential endpoints. So we did a study early on with an AV8 to replace what's missing, G6PC, in the liver of a mouse, complete knockout mouse with GSD1A. And we found that over a couple of weeks, the mice developed normal blood glucose during fasting, and their phenotype improved markedly, where you see these 2-week-old mice just at the time of vector administration. They're very, very small. You can actually appreciate the abdominal swelling from the hepatomegaly. And then by 4 weeks, looking very, very similar, essentially reversing the phenotype, where there's a unaffected and an affected mouse who are almost identical after giving the gene therapy. And of note, these mice don't survive simply with diet beyond weaning. So simply the survival showed a benefit. Now we moved on and studied the various doses of the vector, different AAV serotypes, and we did reveal here that the benefits decreased over time. Between 6 and 12 months, the vector genomes, the vector DNA in the liver dropped by about 10-fold. And similarly, it started lower in kidney, but it dropped between 6 and 12 months. And then we have a nice histochemical stain in the liver that shows where the vector is making G6Pase, the brown cells. Between 6 and 12 months, you can possibly appreciate that the nice brown background here across the liver turns to pink. So generally, we're losing expression between 6 and 12 months, but a few cells are continuing to make the enzyme with a prolonged benefit. It's not unexpected for an episomal vector that's lost related to cell division. This is what was going on. The episomes just sitting in the cells and then gradually being lost. Then on the other hand, a related and similar study from Janice Chu's group at NIH showed that with vector administration at a high dose early in life, again, 2 to 4 weeks of age in infancy of a mouse, they had a correction in the liver where the liver free glucose and glucose 6-phosphate became normal. And this is with as little as 3% of the normal level of G6Pase. And they monitored the mice for up to 90 weeks. So this is all very positive, especially considering that the mice were treated early in life, and we'd expect some loss of benefit. And this has led to a clinical trial, now phase 3, sponsored by Ultragenics. You can see the identifier down in the lower right. You can find this easily on clinicaltrials.gov, but I can't present their data. It's going ahead. Now, there remain some questions about this approach to gene therapy too. On the one hand, groups like Chuck Venditti's group, led by Randy Chandler, found that treating newborn mice with an AAV vector, at least some AAV vectors, there's a long-term risk of the vector integrating and activating an oncogene and producing liver tumors. And so far, this has only been seen in newborn mice. Older mice don't have this problem. It has to do with the change in gene expression, probably the change in chromatin at this oncogene with development. And the thought is that there isn't an analogous gene in humans, so that the risk for this particular toxicity is quite low in humans. But that's just reassurance. We don't know. On the other hand, the study I mentioned for Janice Chu's group in hepatology, showed that mice monitored for over 90 weeks with GSD1 didn't develop any liver tumors. Later, they did more observations. They found that if there was a less than 3% G6Pase activity in this mouse model, they would develop liver tumors, but related to GSD1, not related to the vector. And so it's a mixed picture. Again, risk, benefit have to be considered for each disease. As a way to deal with the loss of benefit from AAV vectors, we've turned to gene editing, what's termed gene or genome editing. And so this shows an example from a few years back. Graduate student Dustin Landau developed in the lab. We collaborated with Charlotte Gersbach, who's a genome editing expert at Duke. At the time, we were using zinc finger nucleases. This top cartoon just shows how a zinc finger nucleus is constructed to cut DNA to create a break at a particular site, in this case, the ROSA 26 gene. And then once there's a double-stranded break, we can deliver a donor vector that has sequence homology to the place the DNA at the break. And at some frequency, then this transgene with its homology will integrate into the chromosome at that spot. So we have the cut, the homology-directed repair, and then an integrated copy of the transgene. So for all those cells, there would be long-term expression of G6Pase. How did this work? Well, in Dustin's experiment, we could see in panel B here for the PCR showing that we could detect the integrated copy. It's not quantitative, but clearly it's there. And that's both in our GSD1 mice and in wild-type littermates. And then without the ZFN, that's a key test to show that the nuclease is driving this. We didn't see very much of the integration of the transgene at all. Then furthermore, we could see at 3 months of age that we had increased G6Pase activity following genome editing in comparison with no zinc finger, no editing. And we had lower glycogen content in the liver. So this showed improved benefits from genome editing. We also had improved survival from the genome editing. So more stable and beneficial effect. And now I'm gonna bring in another facet of GSD1 biology just briefly. We know that there's abnormal autophagy. This is related to the high glycogen and lipids in the liver that accumulate. And autophagy is a process activated during fasting to break down building blocks of the cell, like glycogen, like lipids, for energy. But with GSD1 liver cell permanently full of high glycogen lipids, the cell is sensing a fed state, so autophagy is shut down. And actually shutting down autophagy is damaging in itself. Here's a little bit of the pathway showing how there's an LC32 marker for the autophagosomes that we can measure to measure autophagy. And then as autophagy progresses, these recycling materials are delivered to lysosomes, producing autolysosomes that then break down the glycogen lipids. But this process is impaired in GSD1. We've then done research to activate it, and one activator of autophagy is rapamycin. In an initial study that was beneficial in our knockout mice with GSD1. It lowered the glycogen, although it didn't prolong their survival. And this was all in collaboration with graduate student Ben Farah with Paul again at Duke National University of Singapore Medical School. Here again, showing how shutdown autophagy will impair or worsen GSD1 because lipid droplets and glycogen can't be metabolized. And we know that the glycogen could be broken down in the lysosomes by GAA. So if there were better autophagy, we could lower the glycogen, and that's how rapamycin was working. We then studied another drug, bezafibrate, and bezafibrate is well known for activating fatty acid oxidation for burning fat, including triglycerides. We show that the effect was good in the GSD1A liver, going from lipid-filled hepatocytes, all in red here with the oil red O stain, to a much more normal appearance, just like the wild-type liver from littermates who are also treated with bezafibrate. So we have a drop in the triglycerides from extremely high down to similar to normal in our GSD1 liver just by adding bezafibrate for a week. And this is led by Lauren Waskowitz, Duke grad. And then we combined that with gene therapy, actually genome editing. So the thought was using bezafibrate to make a more normal liver, normal liver cells, and genome editing might work better. And this is all through activating autophagy. Here we are again, our schematic for the ZFN, cutting the ROSA 26 gene in mice with GSD1, and then through homology-directed repair, the G6PC cDNA, the transgene making G6Pase, is integrated and stably treats the mouse liver. So this is a then comparison with and without bezafibrate, and we're staining for G6Pase, and we saw a very, very nice improvement here in the staining in the liver, and also associated with lower glycogen. And this kind of secondary treatment, combination treatment, can really improve the effect of genome editing. This was led by Hai Kang, who was a postdoc in the laboratory. Now, to be complete, I'm mentioning a really recent study from Janice Chu's lab in 2020, and she used the more modern nuclease, the CRISPR-Cas9, and this cut specifically at an R83 variant that had been knocked into this mouse model of GSD1. And R83C is very similar to a common mutation seen in human patients, so it's a good model. The mice had hypoglycemia, including seizures when fasted. And when they were treated with these two vectors, now the one that says Cas9 makes the break, and the other one down here with MG6PC produces a normal mouse DNA to correct the mutation only. And so after this, the mouse would have its normal non-mutant G6PC gene. Now the drawback here, it worked well, but it was in less than 1% of the cells where the homology-directed repair actually fixed the variant. So this too needs further development. And on the third hand, one more thing to mention in GSD1 is mRNA therapy. So it's a kind of gene therapy, isn't it? When when a nucleic acid is used to replace a missing enzyme. And this shows work by the group at Moderna. They packaged the G6PC mRNA with their special lipid encapsulation. They tested 3 different mRNAs, and the glucose increased as shown for these 3 groups here in comparison with the mouse with GSD that got a GFP vector that of course would have no benefit, but that's a good control, giving an irrelevant AAV vector. And the glucose is practically normal through 9 hours of fasting. The other really important point with mRNA is it tends to be degraded. So this is a time course, day 2 through day 9, and much less of the mRNA, much less of the G6Pase protein being produced from the mRNA injection in day 9 and almost gone by day 14. This shows that this kind of treatment would have to be administered frequently to treat the patient. And finally, on GSD1, work in GSD1B. So this is different in that the defect is glucose 6-phosphate transporter. There's also neutropenia. And to treat this, we should treat both the liver and blood cells. But they used an AAV8 vector to show, good correction of hypoglycemia, good correction of liver, without correcting in the blood involvement, the neutropenia. So further development needed here for GSD1B, but certainly there's work ongoing. Now quickly, I'm going to shift gears to Pompe disease. Again, we and others at Duke have done a great deal of work in Pompe disease, including on gene therapy. And we know well that this is a GAA deficiency in the lysosomes that causes glycogen accumulation, especially in the heart and skeletal muscles, and leads to weakness. And in the severe Pompe disease cardiomyopathy, it can currently be treated by enzyme replacement therapy developed by YT Chen's laboratory at Duke and produced by Genzyme, now Sanofi. Our gene therapy is to deliver an AAV8 that has high tropism. It homes to liver. It produces GAA only there because it has a liver-specific promoter. And there are 2 effects. One is the secretion of GAA into blood, where it travels to muscle and heart and is taken up there through a receptor-mediated uptake, as well as there's a unique phenomenon of liver-specific expression where it suppresses immune responses against the GAA. And so we know from ERT that patients who form a high titer of antibodies against GAA won't respond well to therapy. This liver-directed gene therapy actually prevents or suppresses those antibodies. So it could provide perhaps a benefit, especially for infants who would otherwise form antibodies against GAA during ERT. This shows those features. So we did a head-to-head comparison of ERT with AAV, and the GAA was continuously elevated in blood for the gene therapy. The liver's making it all the time. It's like a continuous ERT in terms of its benefits. And then there was a prevention of antibodies against GAA that the mice do form in the face of ERT. This shows over this 1 month experiment of weekly ERT versus a low dose AAV that we had similar benefits. Down here, a trend towards the GAA increasing with the gene therapy, but both treatments clearly lowered the glycogen, and this shows in the heart barely any detectable glycogen after gene therapy, which would be the normal amount of glycogen in the heart, and an equivalent remaining glycogen in the diaphragm for either the weekly ERT or the low dose gene therapy. This told us that potentially gene therapy could replace ERT in Pompe disease. We've gone ahead with a clinical trial, first supported by NIAMS and NCATs at NIH, and then now supported by AskBio. A typical phase 1 design, 3 patients per cohort, 2 doses, medium then higher dose, with primary endpoint being safety, monitoring for adverse events, laboratory testing for things like elevated liver transaminases. Secondary endpoints validated in Pompe disease, including 6-minute walk test and forced vital capacity, as well as some others measuring the GAA activity in the muscle biopsy before and after gene therapy, and GAA activity in the blood, and antibodies against GAA, and the urine biomarker. Duke is a single site, including our clinical program to run the study, and supported by the Glycogen Storage Disease Laboratory to do testing. And just a brief summary for cohort 1. Now we can report, and we're limited in what we can report, but safety indicated by assessment of related adverse events, lack of related serious adverse events, and avoidance of specific defined stopping rules that included things like elevated ALT above a certain level. In this cohort 1, we did not observe ALT elevations that have been associated with the T-cell response to AAV that I mentioned earlier. And we also met criteria for ERT cessation that included a continuous elevation of GAA in the blood after 24 weeks. And so all 3 subjects were able to stop ERT. This is a team effort, and especially it's led by our clinician colleagues, Eddie Smith, Laura Case, and the laboratory, Dr. Bali, Dr. Young, our research coordinators. Ming Zhu has taken this job on and done just incredible work to support our study, and also some unique resources at Duke. We have an Office of Regulatory Affairs and Quality to support communications with FDA. We couldn't have, say, filed the IND and gone ahead with this study without their support. Now, finally, as I near the end of my time, I want to mention another RNA study that's actually ongoing. And so this is also from Moderna. We're a site, we enrolled the first participant for the mRNA study in propionic acidemia. And it works similar to this diagram for MMA, where again, there's an encapsulated RNA that binds to, is taken up by liver cells, and there's production of the normal, in this case, MUT protein to treat MMA, by breaking down the accumulated methylmalonyl-CoA into succinyl-CoA. So the pathway is well understood. What's needed to treat it is well understood, and this is a gene therapy that's actually now in clinical trial. A couple of potential advantages is that there aren't antibodies against these particles, that all patients could be treated. Limitations include the need for treatment every few weeks. I should mention about the propionic acidemia mRNA. It actually includes both mRNAs for both subunits. So there's one drug for both gene variants causing propionic acidemia. But as I mentioned for GSD1, this is transient. The mRNA in the mice lasted for days, not weeks, and the enzyme persisted for almost a week. So it will require, as I mentioned, repeat treatment. But studies ongoing. Our patient continues to get the infusions. So to summarize, a lot of information. I hope you're still with me. I'd say that there are great strides in the field of gene therapy for inborn errors of metabolism, and we have new methods and now successful clinical trials. Let's see, not an approved gene therapy for a classical inborn errors of metabolism, but it's in the works. And along the way, we're learning especially that there are some potentially unexpected toxicities, and also that some of these are dose related, and also potentially some of these will require redesign in a second generation therapy. And linked to that, there are lingering questions, not least of which include cost and reimbursement that will have to be addressed going forward. So here's the team and resources, collaborators, too much to mention, and I'll stop more. I'll stop and if there are any questions, if we have enough time.
Thanks, Dwight. We do have about 5 minutes left where we can take any questions. If anyone has them, they can put them into the chat. But I can start. So given kind of the anticipation that there may be loss of vector or even this tumor risk, are there certain protocols that are being put in place for those human trials or treatments that have already started underway?
Well, yes, especially on the side of safety. So it's becoming more standard to do liver ultrasounds during the follow-up. And really, there hasn't been a report of a drug-related tumor in a clinical trial with AAV. There's been one liver tumor, but this is in a patient with hemophilia who had chronic hepatitis C, and so it was attributable to the hepatitis C infection. So it remains a very much a hypothetical risk, but an important concern, and it's really important to monitor and monitor long term. So FDA is currently requiring 5 years follow-up and reporting for patients involved in clinical trials with AAV. It's only 5 years. It used to be 15 years for all gene therapy, but given that AAV seems to have a good safety profile, and there's more and more data being collected and reported, 5 years is the current follow-up.
All right. Sounds prudent to do that. I see that Dan Scherer has raised his hand, so I will let him ask his question.
Hey, Dwight, can you hear me?
Oh yeah, Dan, hi.
Yeah, hey, great talk. Just outstanding. Really appreciate you doing that. Question regarding messenger RNA therapies. So are there ways, and this may be something, or I'm certain this is something that people are considering, are there ways to subtly modify the messenger RNA bases chemically so that you can still attain protein synthesis, but you can increase the stability of the messenger RNA transcript against nucleases such that the half-life is increased?
Yeah. think there are. I can't, cite those studies. I'm more aware of that kind of chemistry. I'm sure that's why you're asking for siRNAs and for small RNAs that are used for gene suppression usually. But yeah, I mean that holds some potential. The key modification for Moderna has been the encapsulation and the improved delivery to the liver, because historically, say for an RNA therapy or any non-viral therapy, it has tended to go where you don't need it, say the lungs, and be almost entirely taken up there as opposed to liver. So encapsulation, their delivery has really been a breakthrough for them. And and for some in some discussions, they're framing it as, well, RNA therapy is something can be done now, and it could be beneficial when there isn't any other specific treatment for these patients. It may be a bridge for gene therapy. If gene therapy is the future, it may be needed for patients who aren't eligible for gene therapy. It may be a bridge to liver transplant, if that is sort of the definitive care for some conditions to get a stable benefit. And one thing that's being mentioned, I think it's really kind of intriguing, is would there be an mRNA therapy for all cases of inherited disorders of metabolism with hyperammonemia in the newborn period? So you could deliver a cocktail before you have the diagnosis. I think that's a really, really clever and intriguing idea that could improve outcomes.
Yes, that's a great idea. All right, thank you very much.
Mm-hmm. You're welcome. Anyone else brave? We've got 2 minutes, I think. Ask your burning question, even the chat.
Sorry, I got disconnected, but Ronnie asked a question about cost and insurance coverage.
Yeah. So far there's, good precedent for covering these expensive therapies for orphan disorders. And, you look at SMA, and there are 2 approved, therapies they're both DNA based. So I think there's precedent, but the idea of, pricing that is somehow more reasonable, like say payments over the course of time might be more acceptable. And I think there has to be movement for cost containment. So it's a really, really important issue. It has to be developed at the same time as the drugs.
Absolutely, because we want to ensure access after we go through so much effort to develop all of these exciting technologies. So and we can't bankrupt our Medicaid either. Yeah. Right.
There's a question in the chat. What % of the population have pre-existing antibodies to AAV and would be ineligible? And it increases with age. It starts out, say, really the minority, say less than 20%. And then by middle age, it could be as high as 50%. It does vary between serotypes. It's worth mentioning that for AAV5, antibodies seem to be less of an issue, including in the uniQure study in hemophilia. They didn't screen against really significant titers of antibodies against their AAV5 vector, and those patients still responded. So it wasn't the highest titer, but it's surprising, and it's exciting to see that patients with titers against AAV could still be still respond well enough to have a benefit. That may be limited to AAV5. So far, that's true. And then, there are efforts underway to develop immune suppression that'll completely eliminate those antibodies. Those include things like enzymes that cleave immunoglobulins accompanied by plasmapheresis, and then a rapid injection of the vector while the antibodies have been cleared. It's all, under development. Okay. Well, thanks, everyone.